Good day everyone, welcome to the course on biological data analysis and visualization with R. So, we have introduced WGCNA and we have talked about the WGCNA package in the last class, and we have also introduced the data set, and we have talked about the how we process the data set with metadata and gene annotation. So, what we will do is we will continue that analysis now in this class, and we will go into more detailed analysis. So, first I would like to discuss the WGCNA workflow before we actually jump into the analysis, because it should be clear, right, what we are doing and why we are doing it. So, that is something I want to discuss first with the WGCNA workflow, and then we will do the hands on for data loading and normalization. Okay, so, we will continue the normalization part today. Okay, so, in the last class we started with the data loading and we did the first step, which is the drop NA function. So, that is the first step in this workflow. Followed by that, what we have to do is the normalization. Okay, so, what is normalization and why do you need normalization? So, normalization is important because there could be differences among the data sets, the samples that we have talked about. So, these samples, we had 9 gastric cancer samples and 9 normal tissue samples. So, there are differences in these samples, these expression profiles that might arise because of technical variation. Okay. So, there is of course biological difference between normal samples and cancer samples, but there is also technical variation, right. When these samples were prepared, that there would be technical variation that will be reflected in the data. The goal of normalization is to actually remove that technical variation, so that we can study the biological variation, right. So, our focus is biological variation, we want to get rid of the technical variation. So, normalization helps us to do that. Now, what kind of technical variation is introduced? For example, we have PCR amplification, right. So, when you are preparing the cDNA library for next generation sequencing, there might be PCR amplification bias. Okay, so, this is something that is well known that might bias our experiments. We have sampling of transcripts, so that also can bias our data. So, there are a lot of sources of variation, technical variation in our data. So, those are something we need to think about and we need to take care of them. At the sequencing level, we can have different number of reads for each sample, right. Total number of reads for one sample might be 2 million, for another sample might be 2.5 million, and that will again introduce variation, right. So, the sample that have 2.5 million reads, that will appear that genes are higher showing higher expression than the sample that has 2 million reads, right. So, this is something again we need to take care, and normalization does that. There are many different methods of normalization. We will not go into all those details. Some of these have been covered in an earlier course where we actually talked about next generation sequencing data analysis and applications. So, we have talked about different normalization method, what normalization means, that is actually there in much more detail. For normalization in this data set, we will use the DSEC2 package. So, it has a normalization method called median of ratios method. So, again I will not discuss this method itself, because this has been discussed elsewhere. So, followed by that, so once we have normalized the data, so we will take all these data sets, right, the gastric cancer samples and normal samples, we take all these data sets, all the genes, right, we have this big matrix, and we will take that data, we will do the normalization, we will get the normalized data. We will also do something called variance stabilization. Okay. So, this is something that is also important, right. So, for example, the genes that are lowly expressed, they often show very high variance. Okay. It has nothing to do with actual variance, it is something because the genes that are lowly expressed, the effect of sampling is much higher, much stronger in those cases. Okay. So, that means the genes that are lowly expressed, they appear to be more variable. So, this is something we need to do with variance stabilization, and at this stage what we will do is, instead of taking these all 39,000 genes, we will actually do something called gene filtering, and we will choose only the top let us say 10% genes or 5% genes that show the most variation across samples. Right. So, the idea is that the genes that show very after variance stabilization method, the genes that show the most variation, right, top 10% or 5%, they are likely to be different between cancer and normal samples. Okay. But of course, we can actually do this gene filtering in different ways. We can do a proper differential expression analysis and maybe find the list of genes, and then we can also use that filtering for our purpose. Okay. So, but in this class, in this hands on, we will use a very simple filtering. We will not go into the differential expression analysis itself, because that will take a lot of time. Of course, we will not be able to complete that within a short period of time. So, we will use a very simple gene filtering for the genes that show highest variation after we have done the variance stabilization technique. Those will be the genes of interest. Now, once we have done this, the next step will be the soft threshold function. Okay. So, we need to determine what should be the actual threshold. So, this is usually the soft threshold is usually 6 or 12, but again, as you can imagine, this will vary depending on the data set that we are working with. Okay. So, we have to, for our data set, we have to determine this soft threshold. Now, this soft threshold is determined based on whether we get a scale free network, right, at which soft threshold we get the scale free network. So, this is something we will discuss in much more detail when we actually come to this soft threshold part. Once we have determined the soft threshold, we can identify, we can do the network construction or network generation, which is simply identifying the modules of genes that show very high expression correlation. Okay. So, that is the network we have. These are highly connected, means highly correlated genes. So, once we have this network generation, then we can store the network data, of course, for other purposes, or maybe for visualization or some sort of network analysis. So, that is a completely different analysis from here, but those could be applied to this network. The other part we can do is we can identify the modules of interest. This is something what we will follow, is we will want to identify modules of interest in our data. It's what are the modules that show different behavior between gastric cancer and normal tissue, right, because these modules that show different behavior are likely to contain genes that are changed, right, in cancer. Okay. And this, so by that definition, some of these genes could be the driver genes of cancer and maybe could be good targets for drugs. Okay. So, once we identify these modules, we would also look into the gene expression profiles in modules, and we can visualize some of these gene expression profiles and see how the gene expression is changing. Okay. So, in this class, what we will do is we will complete these 3 steps. Okay. So, first we will do the data loading, which we have completed. So, I will simply load the data and we will do the drop NA function, apply the drop NA function, and then we will describe normalization. We will do the normalization and see the normalized data, and then we will do the gene filtering, and after that we will have some visualization. So, at each step we will see, at step by step we will see the data, right, when you are processing, when you are applying certain functions or certain operations, how the data is changing, and of course, we want to visually see, right, how the data looks like after, for example, normalization and gene filtering. Okay. So, this is something we will do now in R and we move on to the R platform, R console, and where we will do this analysis. Okay. So, here is the console, and we have this data. So, we have seen this already. So, we have loaded this library. So, I will simply again check this data. This data should be loaded here, but just to check that data is there, it is there. And we have also applied the, so we can also check the, just to remind you, these are the samples we have 9 tumor samples, right, and so these end with T here, you can see this T RNA-seq, these are 9 samples, and then we have N RNA-seq, so these are the normal samples. Okay. And then the first step that we performed already is the drop NA function, and we introduced this in the data organization, right, when you are talking about data organization with TiDR and DPLYR, we covered many of these functions. Okay. So, we have now dropped any NA value that might be there, and as I said, we want to use the library DSEC2 for normalization, right. So, we can now move on to the normalization part, and the library that we are going to use is the library DSEC2. So, library DSEC2 we have discussed in a lot more detail, but again I can show you very briefly where this DSEC2 is there. This is a bioconductor library, right. Okay. So, this library is actually built for differential gene expression analysis. So, it takes raw data and it actually first normalizes the data by median of ratios method, and then it does the differential expression analysis. So, in here we will not do the differential expression analysis, we will simply take the raw data and do the normalization. Of course, there are different ways of doing normalization, but again we discussed this in our earlier course. We have, for example, the counts per million data. So, this data set actually you can also access the counts per million data. So, here you can see this raw data, if you download, this is the counts per million data. So, we do not want counts per million data. You can also do the transcripts per million. So, TPM normalization. You can do different types of normalization, but there are drawbacks for different types of normalization which we discussed in the earlier course. So, one of the preferred methods is either median of ratios or the TMM normalization. So, here we are using the median of ratios method. Okay, so, this is the normalization method that is implemented in DSEC2. So, we will just take that normalization and we will take the normalized data. Okay. So, that is why we are using this DSEC2 package. We have loaded that package now, and what we need to do is we need to first get this data that we have. So, what we have in the data is, let us see. Okay. So, we want to first remove this gene ID, right, and use the rest of it as the data for normalization. Okay. So, this is how DSEC2 works. So, we are processing these data sets because DSEC2 will take data in a specific format. So, first command is data -1. Right. So, we are removing the first column, which is only the gene name. This will come as metadata, right. So, we will put this in somewhere else, right. So, here is the as matrix we have loaded this data too. We do not need the gene name, right. We will normalize this numeric data. Now, we have converted this to a matrix. The row names will define as the row names of this new data set will be the data gene ID. Okay. So, now you can see, right, instead of the first column being the gene names, we have put the gene name back as row names. Okay. This is something possible as a data frame, not in a table. So, here we are putting this back. Now, why we are doing this? Because DSEC2 will take data in a specific format, right. So, each package has its own input format. So, DSEC2 will take this in that format. So, we have seen the data too now. What we also need is something called an experimental design, right. So, this is something that we have to give to DSEC2 saying these are the samples, right. This is the design of the experiment. Okay. This is something that we do also for differential expression analysis. So, for that we will define this metadata, right. This is a data frame. The sample would be the names of the data file, original data file, the first, so except the first column, right. We have the, so names of those columns will be the sample names, and the type would be, okay, the first columns, right. Except the first column, we have the column names. Okay, so maybe we can break this down into simple things. So, what does this mean? Names data -1. Okay. So, names data -1 is the column names except the first column. Okay. So, that is what we have, or we can simply say names data 2, sorry, names data 2 is not there. Okay. So, names data 1, right. So, except the first column we have these are the sample names. Okay. So, what we are doing is we are putting this as a data frame. We are saying sample is these names, right. So, that should be the sample. The type we have to describe, right. How is this experiment designed, right. We have this tumor versus normal comparison. So, we have to say, okay, these first 9 samples are from tumor samples and next 9 are normal samples. Okay. For that, what we will do is we will extract this T or N from these names. Okay. because we can say okay, T is the tumor and N is the normal sample. So, we want to extract this T and N from the sample name. So, that will define our experimental design, right. So, that experimental design will be stored in this type variable. Of course, you can also manually define this, right, by just saying C repeat these times, right, tumor repeat these times and normal repeat these times. So, this is possible, but this is the easiest way without typing a lot of things. Okay. So, we have defined this, right. So, once we understand what we, so we are okay. So, what I will do is then again use this names. Okay. So, this substring, so this is actually generating this substring at the 9th position. Okay. It is saying it is giving you this T and then okay. So, this is our experimental design, right. So, that is why this part is defining this type value and the sample names, okay. And this we store in the metadata, and what you see is this is how the metadata looks like, the sample and the type, right. So, these samples are tumor samples and these are N samples, of course T or N, so that the DSEC2 package understands, okay, these are part of two different treatments versus something, right. So, two different conditions or treatments, etc. So, that is enough for us to give here. Okay, so, we have created the metadata now for this normalization part, and for DSEC2 this is what we have to do is first we have to define the DSEC data from matrix using this data that we have just generated. So, we will use the data 2, we will give the metadata, and what we have to say is that design is equal to type, right. So, this is how we define the experimental design, okay. Design is type, okay. Round data 2. So, whatever data 2 we have, just take that data and then we have take the metadata, okay. So, this is something we can do. Let us see, we not, if there is any float value, so we can use the round, but otherwise I do not think we need the round command, because DSEC2 takes only integer values, right. So, converting counts to integer mode, right. So, because DSEC2 takes only integer values, if it does not take any float, right. So, this is why we did the round, but in our case it is not required because all data is in integer. Okay, so, this is we have done. Let us see DDS. So, this is a DSEC data set, right. So, to use DSEC2 we have to create a DSEC data set. This is the dimension. Again we have this 39,300 something genes and we have 18 samples, 9 tumor, 9 normal. Okay, so, we now do the DSEC DS. Okay. So, it will take a bit of time. So, here you have the normalization being done. Okay. So, it is saying estimating size factors, etc. So, all sorts of calculations are going on. If you know the algorithm, you probably would know how this works. Anyway, we are not going to go into that because we have discussed how the normalization happens in other codes. So, we have now generated this. So, this is the command DDS DSEC DDS. So, we have now normalized the data. Okay. So, what we have is that, as you can see, more details, row names are the gene names, right. So, we have some values that have been calculated and the column names are the sample names. Okay. And there are 4 things that have been calculated here. Okay. All right. So, we have seen this now. So, what we will do is we will now apply this variance stabilizing transformation, okay, on DDS. Okay. So, this is what the variance stabilization method is doing, right, and we are calculating this variance stabilizing transformation, and based on this we will apply the variance stabilization, we will get the variance stabilized data on the VST data. Okay. And let us see the VST data. Okay. So, maybe check the head, right. Okay. All right. So, we have these variance stabilized data. You can see these now, these are fractions. So, this is the normalized variance stabilized data that we have. Okay. So, we have completed the normalization part and the variance stabilization. So, the next step is the filtering, right. So, as I said, we want to do gene filtering, and this is what we are going to do here. Okay. And for that, we will use this gene filter library. Okay. Okay. So, all these packages are installed, but if not, you now know how to do these installations and how to do these steps. Okay. So, what we are doing is we are calculating this. Okay. So, for this VSD data, we are calculating variance of the rows, right. So, rows are the genes. So, for each gene we are calculating its variance. Okay. And let us see what we get. Okay. So, these are the variation that we have. Okay. And for variance, we see these variance values for each gene. Okay. Now, what we will do is we can actually calculate the quantile, right, what is the 90% value. So, maybe let us see if we can also see summary. Yeah. So, it gives us this first quantile, median, mean, third quantile and maximum value, right. So, what we will do is we will want to select. So, let us see if we can also draw an histogram if it works. Okay. It works, but okay. So, most of the values are here. So, we want to take the top, let us say 10%, okay, top 10% of the genes with the highest variance. Okay. And for that we define this Q90, right, the quantile 90, and this is how we get this the top 10%, right, or the quantile value, right, for this variance that we calculated. We can simply say, right, we have calculated these already here. So, Q90 will give us a number, right. So, this is the 90% value, right, 0.9. So, we can take all these genes that show this above this value, variance above this value, we will select them. Okay. So, that is what we are doing here now. We are choosing genes, right. So, we are choosing this, right, from VSD data, we are choosing genes or rows, right, that is why we have this condition in before the comma. We are choosing these rows where this row variance is greater than Q90 variance, this value 0.9. Okay. And we choose only those rows. Okay. So, we can now see this, right, the dimensions have changed. So, this is the original data, right, after variance stabilization, we have 39,000 of them, and now we have the normalized data, it is about 3,900, right. So, because we have chosen the top ones, top 10%. Okay. So, we will use this normalized data to actually do our analysis. We can of course be more stringent, we can say we will take only top 5%, top 2%, right, because these numbers are still quite huge. So, we can do that again depending on what you are interested in. Okay. So, we have now seen this normalization part. What we want to do is we want to see effect of normalization, right, and variance stabilization on the expression levels. Okay. So, this is something we want to do, and we use something called this plot RLE function. Okay. So, plot RLE function it gives us a very nice value of these distributions, actually how these samples are organized, etc. So, this plot RLE function is part of this RUFSEC package. Again, we have used this package for different types of normalization, we will not talk about that. We will, from this package we will simply take this plot RLE function, okay, and we will plot our data. Okay. So, I just want to show you, right, again this is a bioconductor package, RUFSEC, here it is. So, this is a bioconductor package. Again this is mostly used for normalization, but it has a very nice visualization tool as well, which we can use for our purpose. Okay. So, let us load this RUFSEC. So, let us see. Yeah. So, we have this plot RLE function, you can see this plot RLE, right, and we can use this plotter really for our analysis. Okay. So, let us first load this library rubsec, okay, and we first define the colors. Okay. So, what we are doing is what should be the color of the samples, right, how they will be shown in our case. Okay. So, we can close the histogram first. So, what I am doing is we are saying like we have 9 tumor and 9 normal. So, I am saying orange. So, tumor should be shown in orange color, and the normal tissue samples should be shown in the blue color, right. So, dodger blue here. So, orange and dodger blue. Okay. So, 9 of them. So, that is why we have repeat, right, or replicate orange 9 and replicate dodger blue 9. So, we will see what happens if you see, right. So, we have this 9 orange and 9 dodger blue. So, this is how the colors would be specified. So, we can simply now say color equals to colors, and then this will be automatically applied to the samples. The command is very simple, right. So, we have plot RLE data 2, outline equals to false, okay, and color equals to colors, as I said, right, and these colors will be automatically applied. Okay. So, this is what you see is the original data, right, and as you can see the expression levels, expression values, right, these are actually for the tumor samples, these were actually systematically much higher compared to normal samples. Okay. So, this is something that we notice, which means there was some sort of patch effects, etc. The technical variation that actually was there. Now, let us see how this actually has changed if we now once we have done the normalization, right. So, what I will do, I will take this normalized variance stabilized data, and as you can see now they are kind of much more comparable, right. So, we can see this here, right. If you this is before normalization, right, and you can see that tumor samples actually have higher expression, systematically higher expression compared to normal tissue samples. So, this indicates maybe there is some technical variation, and after normalization what you see is this technical variation has been removed. Of course, there is some difference still, but this variation has been minimized, at least so that we can now look into the biological variation. Otherwise, the technical variation would overwhelm, right, and will mask the effect of biological variation. We can also see the data that we have chosen for our further analysis, right. This is the norm data, right. We have chosen only those genes that show very high variance across samples, and we can see their expression levels, the distribution of this expression of these genes, they are quite comparable across tumor and normal samples. So, which is good. So, which means our normalization and variance stabilization actually worked very nicely. Okay. So, we have done this part, right. So, the next part, so we have now loaded the data, we have done the normalization, we have done the gene filtering, we have done as part of normalization, we have also done the variance stabilization, then we have done the gene filtering part, and then finally, we have compared this visually, right, the data before normalization, the expression data after normalization, and we are kind of okay, right. We are ready to proceed now to actually to the next step, which we will do in the next class. So, let us go back to the presentation, right. So, just to remind you again, so we have completed this data loading, normalization and gene filtering part. In the next class, we will move on to the soft threshold part. So, these are the key functions from the WGCNA package, right. So, until now we have not used the WGCNA package, right. None of these functions required WGCNA package, we used other packages or bioconductor packages. Then the next steps will require WGCNA package, the soft threshold determination. This is a very critical step. We will again do this analysis and again visually check, right, how to pick the right soft threshold value, and then we will do the network generation. This is something that is also important and a key step in the analysis. And then subsequently, we will talk about the identification of modules of interest. We will visually look into the network data, we will actually identify the modules that are interesting to us, and then we will look into the gene expression profiles that are present in these interesting modules, and at the same time we will of course store the network data, which can be utilized for other analysis, for example different network analysis, calculating centrality measures, identifying hub genes, A lot of network analysis tools can be applied. We might not be able to do those because of constraints on time in this course. So, here are the references for some of the packages that we used. Just to summarize, we have loaded the data, and we have also used the metadata for our analysis, right. So, we have defined metadata here, and we have then we have done some pre-processing and normalization. So, normalization included normalization with DSEC2 package as well as variance stabilization, and after this normalization process we identified the genes that showed most variable expression, because we want to proceed with WGCNA analysis only with genes that are of most interest to us. So, we will continue with that in the next class. Thank you.